Welcome to This Explainer. Today, we're diving into a massive, honestly, pretty hidden problem in global agriculture. And more importantly, we're going to unpack the brilliant, microscopic chemical hack that was designed to completely fix it. You know, we rely so heavily on diagnostic tests to tell us the truth about disease. But what actually happens when the very tools we trust to keep our food supply safe start giving us the wrong answers? It's a pretty provocative question, right? Think about it. When a critical medical test lies, what happens? Normally, when we take a test, we just expect a simple binary answer. You're either positive or negative, safe or infected. But in the real world, biological diagnostics are incredibly messy. And when a test lies, it creates this massive ripple effect of consequences, especially in large-scale livestock farming. So to really understand the real-world stakes of inaccurate disease testing, we're going to look at one specific, devastating disease that ravages animal populations all over the globe. Take brucellosis, for example. It is a highly infectious zoonotic disease caused by Brucella organisms. In livestock, like cattle and buffaloes, it causes absolutely severe reproductive issues. We're talking about widespread abortions, placentitis, and just massive herd infections. And the economic toll? It's literally staggering. Just to put this into perspective, in India alone, the livestock sector suffers an estimated $58.8 million in economic losses every single year, simply because of brucellosis. It's a full-blown endemic crisis, and catching it early is really the only way to stop it in its tracks. But here's the stark diagnostic dilemma we actually face on the ground. The standard tests, they regularly throw up false negatives and false positives. Now, a false negative is when a test tells you an animal is perfectly healthy, but it's actually infected. That leaves a highly infectious animal hidden right in the middle of a herd, silently spreading the disease to all the others. A false positive, on the flip side, claims an animal is infected when it is actually totally healthy. The tragic result there is the unnecessary culling or slaughtering of highly productive, perfectly safe animals. Both of these errors are just catastrophic for farmers. Okay, so to understand how scientists are actually fixing this, we first need to define the core mechanic of the most common rapid screening tests used out in the field. It's a process called agglutination. Simply put, agglutination is just the clumping of antigenic particles by antibodies. See, when an animal gets infected, its immune system naturally produces antibodies. In a diagnostic test, scientists will mix the animal's blood serum with killed disease particles. Those are the antigens. If the antibodies are present, they latch onto the antigens and basically clump them together into a visible mass. If you see clumps, the animal is positive. Simple enough, right? Well, not quite. The current gold standard rapid test, the Rose Bengal Plate Test, or RBPT, has some serious microscopic blind spots. Here is why these conventional tests fail us. False negatives often occur when an animal has a really low concentration, or titer, of antibodies. The complexes that form are just way too small. They create these invisible clumps that the human eye completely misses. This is sometimes related to what's called the prozone effect. On the other hand, false positives happen when improper mixing or things like fibrin clots create fake clumps. The particles stick together nonspecifically, which completely mimics a positive result. But enter our micro-solution, the Superagglutination Test, developed by researchers Mohan, Saxena, and Papavitar Kar. This is an incredibly elegant and, honestly, super cost-effective modification to the standard slide agglutination test. Instead of completely reinventing the wheel, they just figured out a chemical hack to amplify the visibility of the test. It's designed specifically to expose those diagnostic blind spots that cause both the false positives and the false negatives. This hack relies on a brilliant four-step procedure. Step 1, you add the standard RBPT antigen, which naturally comes stained a bright pink color. Step 2, and this is a really crucial modification, you take the animal's serum and stain its antibodies with kumacine blue dye. Step 3, you add biotinylated antiglobulin. And finally, for step 4, you add streptavidin or avidin. By cleverly using this specific color coding and adding these targeted binding agents, the true test results are basically going to jump right off the slide. Behind the scenes, this whole thing relies on a molecular concept that essentially acts like molecular Velcro. The antiglobulin binds to the original antibody-antigen clumps. Now, because this antiglobulin is attached to biotin, and avidin has a remarkably strong affinity for biotin with 4 distinct binding sites per molecule, it acts just like a chemical magnifying glass. It physically cross-links and literally glues all those tiny invisible antibody clumps together into massive, interconnected, highly visible structures. And the visual proof? It is undeniable.
With a standard RBP test, you can barely see the clumping. It's subtle, the color is uniform, and it's incredibly easy to misinterpret. But with the superagglutination test, you get a massive, unmistakable clump. The difference is absolutely night and day. This chemical amplification successfully turns microscopic, invisible reactions into undeniable physical evidence.
Now, we absolutely have to talk about the genius of using two colors here. Remember how the antigen is pink and we stained the serum antibodies blue? Well, this instantly exposes false positives. If improper mixing creates a fake clump of just the antigen, the clump is only going to be pink. If it's a fake clump of just the serum, it'll only be blue. But a true positive agglutinate, where the antibody has specifically found and bound to its target antigen, will clearly display both blue and pink colors under magnification. Fake clumps will only ever show up as a single color. It completely, 100% removes the guesswork. Okay, let's zoom back out from the microscope for a second and look at the hard data. We need to see how superagglutination actually performs in a rigorous, head-to-head battle against conventional diagnostic tests. Researchers tested 200 bovine serum samples, taking them from both healthy animals and herds suspected of having brucellosis. They did this to compare the new superagglutination test directly against the traditional RBPT, the standard Tube Agglutination Test, or STAT, ELISA, and the Complement Fixation Test. The numbers back up exactly what we just described under the microscope. In a direct clump size comparison, the conventional RBPT yielded a mean clump size of 1534. But the superagglutination test, it yielded a mean size of 2752. That means the new test creates clumps that are roughly 1.8 times bigger. This difference is highly statistically significant, and it proves exactly why these results are so vastly easier to detect, even with just the naked eye or a really simple hand lens. Furthermore, the statistical victory just continues when we look at the test's overall sensitivity. So while the traditional RBPT is widely used, it peaked at a sensitivity of 93.33% in this study. The superagglutination test pushed that up to 95.88%. It successfully detects early infections that the older tests completely miss, while also maintaining an incredibly high negative predictive value of nearly 96%. In real-world terms, that improved sensitivity and specificity has a profound impact. Out of all the samples tested, the superagglutination test successfully caught 27 false negatives that had been completely missed by combinations of the RBPT, STAT, and even ELISA tests. It also successfully identified 9 false positives that had tripped up those exact same traditional tests. That is 27 highly infectious animals flagged instead of being left in the herd, and 9 perfectly healthy animals saved from unnecessary slaughter. There's a quote from the researchers that I think perfectly captures the global utility of this innovation. They noted, "The new modifications do not require any extra equipment and they do not cost much. This isn't some complex solution that requires millions of dollars of advanced laboratory infrastructure. It only requires a few microliters of inexpensive dye, some diluted biotin-avidin antiglobulin, and avidin. It gives you a highly accurate diagnosis in under 5 minutes right out in the field. That makes it an unbelievably powerful tool for developing nations and remote agricultural areas." So we're going to wrap up this explainer with a provocative question for you to consider. The core principles of superagglutination, amplifying clump size with avidin biotin and confirming specificity with that brilliant dual-color staining, they aren't just limited to brucellosis. What other global diagnostic failures could a simple chemical hack like this solve next? By totally rethinking the fundamentals of how we actually visualize microscopic reactions, this elegant, low-cost approach leaves us wondering how it could revolutionize rapid testing for a whole host of other infectious diseases in both animals and humans all over the world. Thanks so much for joining me for this explainer, and remember to always keep questioning the science behind the results.